Assalamualaikum warahmatullahi wabarakatuh and hello. My name is Mira Azhana Jiha binti Zainurin and my partner Nur Ira Izati binti Saifuddin. We are postgraduate students from University Pendidikan Sultan Idris. Our group, represented by the name Me Ira from the Chemistry Department, Faculty of Science and Mathematics, University Pendidikan Sultan Idris, will be presenting about our innovation called Waste Fish Scales as Scaffold for Bone Tissue Engineering. In this video, we will be talking about the importance of tissue engineering in medical field, problem statement and issues arise with current bone scaffold available commercially, our objective as well as the innovation we came up with and its analysis. In addition, the benefits of the innovation will also be presented, and the conclusion. According to Tonelli et al., tissue engineering is an important field of regenerative medicine for tissue repair. In this study, we will be focusing on bone tissue repair by utilizing the scaffold that is needed to cure bone defects caused by disease such as osteoporosis or occurred from the accident. In order to achieve the goal of bone tissue reconstruction, scaffold must meet some specific requirements such as high porosity and great biodegradability. The example of synthetic material used as scaffold that is already available commercially are polylactic acid (PLA), polyglycolic acid (PGA), and polycaprolactone (PCL). The synthetic materials that is available as bone scaffold associated with disadvantages such as high production cost due to the usage of variety of chemicals. Meanwhile, bone scaffold made up of the derivation from natural materials such as porcine and bovine contribute to the rising of halal issue. According to the diagram, the scaffold derived from natural or biological materials showcase the highest demand in tissue engineering market in compared to synthetic and other scaffold material during the year 2019. It is predicted to top the chart until year 2027. On the other hand, according to the Food and Agriculture Organization (FAO), Malaysia is one of the top fish consuming countries in Asia, which is above 40 kg per capita per year. And according to Evelyn Tay, an average Malaysian consumed 59 kg per year in 2015 compared to 23 kg in 1975 based on the fish consumption trend. The trend is based on the data taken from national fish consumption compared with Malaysian population. Therefore, our objective is to produce the scaffold for bone tissue engineering from natural material in which waste fish kill. The tilapia fish has been chosen in this study. The main components of the fish are the head, viscera, scales, fillet, and bone. The fish scales that present up to 3% of the fish usually categorized as a waste, and our study made the best out of it by utilizing the fish scale as the starting material. Junichi 2005 stated that the fish scale has two major layers, which are the outer layer consisting of hydroxyapatite and collagen, and inner layer consisting of only collagen. Meanwhile, Huang 2007 denoted that fish scale composed of 55% of hydroxyapatite and 45% of collagen. Hydroxyapatite is the ceramic materials that is similar to animal and human bone. Hydroxyapatite can be derived from the synthesis from chemicals, mammalian bones such as porcine and bovine, and marine sources such as shell. Collagen have been used in tissue engineering scaffold preparation. Most of the commercial collagen is from animal sources. Marine collagen is the best alternative to commercial collagen due to its halal status and low production cost. The fabrication of porous collagen hydroxyapatite scaffold from fish scales is shown in the step-by-step procedure. The scales of tilapia fish was washed off until it is clean and then dried up. The scale was then grinded mechanically until it turns into fine powder. The powder was then dissolved in water and cast into plastic mold. The mold was left to freeze and went through freeze-dry process to be converted into porous scaffold. The collagen hydroxyapatite porous scaffold is then went through characterization and analysis. The characterization and analysis that has been done was scanning electron microscope (SEM) analysis, thermogravimetric TGA analysis, X-ray diffraction XRD analysis, cell attachment study, and cytotoxicity study. The SEM micrograph at 500 micrometers showcased the morphology of the 3D porous scaffolds of collagen hydroxyapatite composite, revealing significant porous structure. Based on the graph obtained from the TGA analysis, it is confirmed that there are the presence of both collagen and hydroxyapatite. When the fish scale is heated up, the organic material in which the collagen is burned off and used up as accordance to the red line from the graph. The inorganic material, which is hydroxyapatite, will remain. Therefore, we are able to calculate the percentage of both materials. The XRD analysis is a rapid analytical technique primarily used for phase identification of a crystalline material. The hydroxyapatite phase can be found on the XRD pattern. The peak of the highest intensity for hydroxyapatite was obtained at 2 theta value of 32.19 degree corresponding to 112 plane. The cell attachment study has been done in order to observe the cell growth on the scaffolds. As you can see from the images, we developed a special foam scaffold for tissue culture purpose, and that is the reason for it to appear as flat structure instead of porous structure. This also helps to observe the cell growth more clearly. At 24 hours, the fibroblast cell started to grow on top of the scaffold. At 48 hours, the cell keeps growing at a fast rate, covering the major parts of the scaffold. The observation at 72 hours indicated that the cell is already confluence, covering all parts of the scaffold. We can conclude that the cell grow at a fast rate and have a great growth rate. In order to determine biocompatibility of collagen hydroxyapatite composite, in vitro cytotoxicity test was conducted on the composite sample. Based on the standard protocol, when the cell viability obtained is above 80% after the analysis, then the sample is considered as non-cytotoxicity or biocompatible material. In this study, the cells were treated with the leaching solution of collagen hydroxyapatite composite at different concentration of 25, 100 and 200 mg per milliliter. The overall results demonstrated that the cell exposed to 24 hours of extract showed a high percentage of cell viability, which was above 80% for all composites. These results demonstrated that collagen hydroxyapatite composite was biocompatible since the cell viability result was above 80% for all tested concentration. There are plenty of benefits from this innovation. First and foremost, the price of this natural scaffold is cheaper because the raw material used is the fish scales. The manufacturing cost also reduced drastically in compared to synthetic scaffold that use a variety of chemicals. In addition, the scaffold is safe for Muslim consumer as the source of material and the process is in compliance with halal requirement. Next, it is beneficial to those in fish industries as they are able to generate extra income due to the demand for the fish scale will be increased. Furthermore, this innovation contributes a lot to the well-being of the environment as we made a good use of the aquatic waste. The aquatic pollution or the pollution in general will be able to reduce rapidly. In conclusion, a simple and economical technique was successfully developed to produce porous 3D collagen hydroxyapatite scaffold. Both collagen and hydroxyapatite from fish scale have a great potential to replace the synthetic materials that is available commercially for biomedical application and bring a lot of benefits. Thank you for watching.